welcome to our Sano seminar today. Today we have a very special Sano seminar because we have more than one speaker. We will have 5 speakers, and these speakers are ladies who decided to join Sano for a kind of scholarship for girls in science, in technology, organized by Sano, and they are working still in different Sano teams, and objective is just to present results of research up to now, and a kind of perspectives what will be in the future. So I think that we will start with, as I understand, with Emilia Stacharczak, yes? Emilia was working together with a team which is involved in structural and functional genetics. And the leader of this team is Tomek Kościółek. Let's go directly to Emilia and presentation of what Emilia has done. So Emilia, screen is yours.
I will start by introducing myself. So I'm Emilia. And let's start with some facts about me. So I've always enjoyed learning, especially biology and chemistry. Like probably most of us, sometimes I like to bury myself in studying for the entire evening because discovering new things can be just fascinating. And as a kid, I wanted to be a scientist. Normal dream for a kid, right? Later, when I was in school, I thought I would become a doctor. But when I really thought about it, I didn't feel like it was my destiny. So I started to looking for other paths related to my interests. And that's how I started studying biomedical engineering. Not even knowing then that engineering studies involve a lot of mathematics, physics, and programming. Despite that, I quickly started to like learning about new technologies and about using them in medicine. And soon I'm starting a master's degree in data science, and I plan to just deepen my knowledge, gain experience, and see where it takes me. So far, I really like where my decisions have taken me, and I'm curious about what the future will bring both in science and in my career. And maybe someday I'll become a doctor, as I was thinking, but not just the way I imagined when I was in school. Okay, my interest, as I said, focus on science, and I was looking for a combination of my interest in biology and chemistry with the engineering skills I earned during my studies, and I found it in bioinformatics and computational medicine, and these are the directions in which I would like to develop. But outside of science, in my free time, I like to do sports. I even belong to an academic athletics club, and I participate in running events, but I do this only for fun, so I don't have any great achievements. Yes, I also highly enjoy traveling and science fiction movies, books, and games, especially The Witcher, which I'm really into right now. And it's time to finally say something about my project at Sano. It is about identification of secreted bacterial proteins from the human gut microbiome for host microbiome interaction predictions. As it has been said, I do this in structural and functional genomics group under the supervision of PhD Tomasz Kościółek and PhD Alicja Wojciechowska. Why we do this? Because research shows the increasing influence of microbiome on human well-being and its connection to various diseases, even those that not appear to be related to the gut, including neurodegenerative ones. And understanding of biomolecular mechanism between the gut microbiome and the host would be a major step into better diagnostics and to development of targeted therapies. But of course, this is not so easy to achieve. So the general goals of the project are to identify suitable existing programs and algorithms, and we chose Rosetta Fold 2 Lite, the optimized neural network for protein-protein interaction prediction. But during the test, it turned out that amino acid sequence it's not sufficient, as the network based on paired MSA, which means similar proteins of both proteins from a pair originating from the same species. And these aspects makes it very difficult to use this neural network on interspecies pairs, like we wanted to use this for bacterial proteins secreted by the gut microbiome with human proteins. So this may not be the best tool in this case, and we will look for alternatives. But for now, we are focused on analyzing the UHGP dataset. This is the extensive dataset containing over 13 million proteins secreted by the gut microbiome. And I am currently working on predicting the subcellular localization of these proteins and analyzing their taxonomy. And what we would really like to do is to predict and analyze protein-protein interaction of human proteins and UHGP proteins. And what we would like to achieve is a database of secreted microbial proteins and their putative interaction with host proteins. Or at least getting closer to it, learn about the tools, possibilities, and limitations to get to the dataset. And it is important to say that such a dataset would only be a basis for planning laboratory tests. So the path from receiving the dataset to creating drug is also long. But for me, this project taught me a lot about the practical use of bioinformatics tools, which is very valuable for me. Okay, so one more slide about other projects I'm involved in, because as my friends say, I like to do side quests. So I'm also a member of AGH Space System Rover Club, where we are conducting research planning, searching and analyzing scientific literature, formulating and testing hypotheses, and preparing scientific reports. I am working as an intern in startup called ShapeCare, where I collaborate with a team of programmers to develop innovative computer vision solution for rehabilitation. My tasks there are mainly preparing data sets for model training and analyzing human movements from videos using AI tools. And I also did a few smaller projects during my studies. And that's all from me. Thank you for your attention.
William, many thanks for presenting your project. Well, can you tell us when you have started this project and how long how long it will last?
I started this project in October, and it will last until April, like a half of April.
And do you have any plans about what to do with the results, about publication, presentation, or something like this?
We don't have very specific plans because we don't really know what exactly we will obtain. If the data set, we will gonna obtain the data set I was talking about, or only we will learn about the possibilities and how many limitations we're gonna get through the journey, because we already had one with the neural network, because we didn't expect that we will have to generate PMSA, which I said we was thinking that amino acid sequence will be sufficient. So we will see what we will obtain, how far we will go.
Many thanks. Well, I think that you can count on a kind of support from everybody in Sano, and well, not only colleagues who are always here, but you can also think about support from people like Wiesław, for example, who is among attendees of this seminar. So many thanks for being brave enough to be the first presenting these results, and in public way. Thank you very much. Well, as I understand, the next presenter will be Elena Gomez del Pozo. Yes, yes. Oh, it's you.
Yeah.
Okay, so Elena is in Sano. I think that it is the second or third month of
Yeah, almost first already, actually. I'm starting the fourth right now, actually.
and well, you are working in the team X and scale data and computing, yes?
Yeah.
And I don't know if Maciek has managed to come here after meeting with person, no?
Yeah,
But almost.
Should I say something?
Well, about the Lena, it would be good.
I think I'll just introduce this summary.
Slides.
Yeah.
So go on.
Okay. Well, hi everybody. I'm Elena Gomez del Pozo, as he very well pronounced. And I'm a physics engineer here at Sano. Okay, so physics engineering, it's not so popular. It's actually, I'm one of, I think, the third generation of my studies in Carlos III University of Madrid. I'm from Madrid and I studied in Madrid. And the physics engineering degree is kind of a mix of nanophysics, biophysics, particle physics, and quantum physics, all very physical and theoretical. But they wanted to, for us to be able to implement it on some product or have some application for it, which has a lot of applications, by the way. And so we had also fluid mechanics, heat transfer, signals, and material engineering, which is a very engineering point of view to all of these previous subjects. So, for example, it would be like signals or systems with nanophysics. So we got nanoelectronics and nanophotonics. So you could see you could see all the applications that has. If not, you can ask later. So it was not easy at all to choose this degree, by the way. I have a lot of interest, and so that you get an idea, the second option was philosophy. So I was really lost when I was 17. But anyways, I got physics engineering because I was very curious about how the world works and how to kind of improve it in some way. If that sounds a little cheesy, I'm sorry, but the plan was to learn something that I couldn't learn any other way, and I thought physics engineering was pretty solid for that. So it was 4 years because in Spain we got a bachelor's of 4 years. In the meantime, I had to do an internship. I did it in a factory of Tetra Pak. It's like for little cartons, like milk cartons. So I was working in that. Then I gave some seminars by the end of it to kind of motivate students in high schools to also join engineering.
Yeah.
That was fun. I was also part of a students association. It's a European thing, Board European Students of Technology, and it has centers in a lot of cities in Europe. So we got to travel a lot and make some engineering competitions, and it was very collaborative. Collaborative. That was very fun. I was in this board. This is everyone from Madrid. I also did my exchange 1 year in South Korea, Seoul. It was also super fun. I learned a lot. I really like Korean culture now, although also to choose the destination, you may wonder like, why Korea, right? I wanted to go to somewhere that I wouldn't like, or it would be very hard to go to live and like work and establish myself. So I thought Europe, here I am. Like I thought it was a little bit easier to just go without the support of uni. But Korea, I was like, wow, I probably won't get the chance to live in Korea if it's not through an exchange. So that's why I chose Korea. This is me in the North Korea border. They have like this kind of museum that you can visit. And then this is also me in like a hackathon that we won for ESA. This is my team. Yeah, it was very fun. It was also towards the end of the degree, and we did some space engineering kind of. So space weather, which is the solar storms that get thrown at Earth and cause the auroras. We had to predict. Well, it's very hard to predict because it's kind of like earthquakes. They don't really have a pattern, but we got to program some pretty nice alarm system for the operators of the satellites so that when they get the solar storm, they can close the solar arrays or do some kind of procedure to not get super harmed and fried basically by the solar storm. So I did a lot of things. I'm very proud of all the things that I got to do. And then I had to choose my bachelor thesis, right? There were a lot of things that we could choose from because our degree is quite broad. But since I was in Korea and sunscreens are a huge thing there, I tried to study how the human skin reflects the sun with and without the sunscreen. So I was trying to find a pattern that wouldn't be super reliant on skin melanin because it's very typical that like solar protection factor, for example, SPF 50, SPF 30, it really is based on fair skin, like type 1 would be. There's 5 types, and it gets darker towards the fifth. So it's based on type 1. And I wanted to find a physical way to measure if you have sunscreen or not so that it wouldn't be so skin based on your type of skin. So that was the study. That's my arm. I just asked a lot of people that I know to put their arms in this same spot to measure with and without sunscreen. And I could see the difference in reflectance, and that was a way to measure if they had sunscreen or not. That was my bachelor thesis, kind of related to where I am pointing towards. I hope you see the vision. Now I'm here in Sano. I finished my degree and I wanted a short period job to try to go more into the medical physics part of the field because the field is very broad. And so here I am doing cardiovascular system zero-dimensional models. Actually, the models were already there. What I'm trying to do, these are the models. So this is the heart, and this would be the lungs, and this is the whole body, right? It gets circulation all over. And what people before me did is have a simulation of this like an electrical circuit. So instead of heart valves, for example, you have capacitors with changing capacitance. So they like contract and extend, like capacitors with changing capacitance. And all the veins and arteries would be just like cables with resistors. And yeah, that's pretty much, I'm not gonna go super into well, you also have diodes as valves. Anyway, this is supposed to resemble the cardiovascular system. And what I'm actually doing is taking all these differential equations that describe how the electrical circuit is going. So you really don't have to know about cardiovascular system as long as you know about electrical systems. These are the equations that govern the electrical system. and they have relationships like voltage would be pressure, capacitance would be volume, those kind of relationships. And what I'm trying to do is, okay, so this model is way more simpler than this model, right? This one has many more parameters and it's more complex. It's better for describing the actual cardiovascular system, but this one has way less parameters, and it's very nice for personalization, the digital twin. So the point is to, with this one, which is simpler and has less parameters, introduce a physically informed neural network so that it approaches the more complex one without adding the parameters. So we would just infuse complexity without the parameters. That this is the infusing. So it's just literally adding values to the derivatives in one cycle so that the shape of the function changes. This is a more visual way to see it. The green one would be the more complex one. As you can see, it has like sharper changes in the derivatives. And the red and blue ones are, the red is the most simple, the blue one is like in between. And what we're trying to do is add or subtract to the derivative so that it approaches the more complex one. So that's what it says here. And yeah, that's what I like, and that's what I'm trying to point towards. So I'm trying to go into a master's in medical physics or something similar, trying to apply physics to the human body, like with nanoparticles or simulations, or there's a lot of things that you can do. But I'm really interested in the human body because of the proximity that it has to me, basically, because there's a lot of applications of my bachelor's to nuclear reactors or space. I was really interested in space, but I just feel like everything is so far from my actual life. I feel like the human body, I feel way more connected. I'm way more motivated by all the results that I get. So there's one master's that I'm very interested in, and it's about the human body in extreme conditions. So for example, like skydiving, super high speeds, you don't have oxygen, like it's a huge thing. Scuba diving, also super high pressures. You have to go little by little or your ear will explode. That kind of extreme environments that you see here, especially doing sports, I feel like it's pretty extreme. So I'm really interested in studying how the human body can adapt to all these situations and still survive. Like it's actually pretty improbable that we are surviving to all these conditions. And I'm really interested in also engineering gadgets so that we can even survive better and longer. And yeah, that's me. And that's the end. I'm happy to meet you.
Thank you very much.
Thank you.
So I understand that now you are trying to understand what is going on, how to simulate flow of blood with, let's say, a kind of analogous system.
Yeah.
Well, equations are the same, so it's.
Yeah, I mean, it's all math in the end. It has a very fancy name, but it's all math. Hopefully it works. I'm very faithful that it would work. And we are going to present also in this conference in Zakopane. So let's see if that also works. But yeah, hopefully we can share some nice results and yeah, there's actually a bachelor team of AGH that did their thesis in kind of these concepts of infusing neural network to the derivatives. So I'm just taking the project and developing it further. That's the plan.
Okay, many thanks.
Okay.
So thank you very much. And now we have a third speaker. Yes. This third speaker is Zofia Seweryńska, who up to now was completed study in automation and robotics, yes? And it was here in Krakow or in Silesia?
In the Silesian University of
The
Technology.
University. And now you are working towards your Master of Science degree, also in
Yes.
in Silesia University, and it is in data science. Yes.
Yes, correct.
And for this reason, you have been you decided to work on data which are from EEG. Yes, here.
Sure, not particularly in EEG, but I will talk a little bit about it later. Yeah.
Okay, so floor and screen yours.
Okay, I will just minimize everything. I think now it's correct. Yeah. Okay, so welcome, and I would like to show you my project here at Sano, also to talk a little bit about myself and other projects that I'm interested in. So my name is Zofia Severyńska, and I'm a data scientist. So a quick introduction. I live in Gliwice in Poland, so in the Silesian part of Poland, and I have finished my bachelor's degree in automatics and robotics. And as my bachelor's thesis, I did a combination of arts and robotics. So I made actually a kinetic sculpture that was moving by using some of the robotics mechanism. And now I'm doing my master's degree in also Silesian University of Technology, and it's in the field of data science. So I switched totally my degree from my bachelor's towards more data-driven algorithms. And now I'm currently working here at Sano and also in the National Institute of Oncology in Gliwice. So my project here at Sano was held under the supervision of Professor Jakub Barbasz. And now I will talk a little bit about this project. So first of all, we were focused on the aerobatics and about the pilots themselves. So we want to help them out to become safer when they are flying into the air. So we decided to take this project and help them out. So here we can see a plot that is telling us that most of the accidents are actually happening because of the loss of control of the pilots. So what is this loss of control? It's for example, the main part, so the loss of consciousness and spatial disorientation. But what are the factors that are there? Are the loss of focus. These are fatigue, dehydration, for example, also the fear before pre-flight checks, and the pressure. So we have a lot of factors that contribute to maybe happening to these accidents in the air. And we want to help the pilots to, of course, don't have these accidents because we don't want them to die or anything bad happening to them. So we decided to somehow track their vitals in order to, for example, tell them later, okay, today you can fly into the air, or okay, maybe you shouldn't fly in that day. So we use the Garmin watches and use the data from it, which is in the form of fit files. And we analyze the sleep data of those pilots, the stress data, the HRV data, and HR data. So by doing so, we can, for example, have information about each of those days that they were wearing this watch. And if they, for example, had a good sleep or a bad sleep, or maybe the day before the flight, it was very stressful for them, and so on. So how we did it? We have a schema right here. So first we are taking the wearable data. So we track the sleep, the stress, and so on, as I said. Then we performed a quick test for the reflex to see the brain speed and to see what is the reflex of this person in particular day. Then we were doing a personalization. So for each of the pilots and people, we make some kind of unique physical baseline. And then we perform the AI analysis. So we use a residual neural network to later on merge the physical and mental fatigue. And as an output of this neural network, we have a daily score. So we receive a score of some so-called readiness. And by that, we can have later the final decision if someone should go into the air or maybe should stay low. So now I will just briefly show you a couple of slides how it looks like. And also, sorry, it's not in English yet. It's in Polish, but I will just briefly talk what is here. So we made a web app that is supposed to let the pilots and also other person, if also want to contribute to it, they can also sign up. And it's supposed to analyze the files of those persons who are using the Garmin watches. So we have here the main page and also the login page, but it's not that important. Here we have some sub pages. So we can, for example, track all of the days, as we can see on the left. So we can choose a day and then, for example, track using the diagrams, the sleep data, the stress data, and so on. And we have also this very simple reflex game, which is just working in a way that we change the color from black to blue. And then we measure how fast the person was clicking to see the reflex of this person. And we do 5 tries of this, and then we take the median. And by that, we have a score for the day that the person was doing this reflex game. And the main page is looking like this. So we have the sleep, we have the HRV, and the reflex game. And we have also the most important fact place right there is on the left corner. So it is the readiness score. So as I mentioned before, it is compiled and outputted from the neural network itself. So we want to have it as high as possible to know that the pilot is able to fly that day. And some future plans here at Sano. So this project is pretty nice, like at this moment, but it's not fully like developed yet because we are still waiting for the virtual machine to have later and cloud access to make it like available to everyone everywhere. And we also then, after getting some more users and so on, we will have big data. So it would be better for our neural network to work and to make some better predictions. And also we want to make it more secure and to make it safer. So now I will talk a bit about the projects outside of Sano. So my interests don't align perfectly with this project, but I also like learned a lot, and I'm very happy that I had this opportunity to be here at this project. So in my studies, I'm mostly working on the glioma segmentation, and I'm more towards the deep learning part of it. And also now in the National Institute of Oncology, I'm more working in the statistical part about the gliomas. So we are trying to distinguish between 2 types of gliomas, the wild type and the mutant type, by analyzing the 2Hg oncometabolites. So I'm very interested in the brain area. I'm interested in the statistics and mostly into the healthcare itself. And some of my past projects also were about the EEG data. So I also worked with the time domain. And as it is for the future plans, so I would want to maybe learn more about the small things inside of us or inside of the brain. So for example, the neurons, the cells and so on, and maybe make a project with something like this to get to know it better. Also, I'm interested in the space, so I'm making some projects also with that. And maybe in the future, I would want to maybe teach some other students, and so make a PhD or something like this. So yeah, these are my future plans, and that's all for today. So thank you.
Thank you very much. Well, as I understand, your research is almost completed, yes? So it's, do you have any plans about publication of this? Or?
We still, as I was talking with Jakub Barbasz about it, he wants to make a patent from it. So I think we need to wait for the patent and then do a publication.
Ah.
But I'm not sure.
So in this term, it's interesting.
Yes, I think so.
Yeah, yes, it's very interesting. Well, okay, and I hope that traveling between Gliwice and Krakow is not
It's
so.
no, like mostly I work remotely now, and whenever Jakub wanted to meet in person, we just were coming and meeting here. But it wasn't a problem. No, like yeah, it was okay.
So many thanks for presenting this. Thank you very much.
Thank you.
Well, so now we have a speaker number 4 in this show. It's Zofia Sikorska. And Zofia is at that moment, if I'm not wrong, you are just at the bachelor level of your studies in bioinformatics at Jagiellonian University. Yes, but you have a lot of different interests. So, well, we have Jan here, so maybe Jan.
Yeah, but we have also Jamal here, who is actually supervising the project because this is part of his grant. So, Jamal, if you could.
Thanks for promoting.
Introduce somehow.
Chain of command.
Yeah, Zosia is last year.
Oh, you have learned this.
Yes. I mean, I got corrected quite a lot of times in these 3 years that I spent here. So yeah, now I address people with their softer name, let's say.
Reinforcement.
Yeah. So Zosia is last year bachelor's degree student at Bioinformatics Department of Jagiellonian University. She's working with us in the project where we explore the interaction between oculomotor, neural, and behavioral dynamics after brain stroke. Her specific task for the project is to extract eye movements from fMRI images using deep learning methods. Because we already have some linear methods working up to a certain degree. She's working with deep learning methods to see if we can improve the accuracy of the data we extract, features we extract. And she's writing her bachelor's thesis with this project. And she used the outputs of her current progress to present a poster in the Neurons conference in Krakow. And floor is yours.
Yes.
Hello everyone, my name is Zofia, and I will be talking today about decoding the eye movements from fMRI while people do absolutely nothing, which can be sometimes hard, and using AI to like predict these eye movements without throwing my computer out of the window. So I will start with the bigger picture, the overview. Why are we doing this and what are we doing? Then I will briefly talk about my attempts, the final breakthrough, and at the very end, what I am going to do next, and a little bit about me. So starting with the big picture, why do we even want to decode the eye movements from MRI scans? As you already know, I'm under Jamal's project, and we know then in strokes, people have glitches in oculomotor system. And thanks to decoding the eye movements from the resting state, we can map how the brain reorganizes itself, and it can be a game changer in further diagnosis. And there's the catch, because we already have an amazing tools like deep MRI, which is amazing for active tasks such as smooth pursuit. But during the resting state, the eye movements are really tiny. It's like ±1 degree. And when we feed this tiny whisper of movements into our model, it basically hallucinates, which is not good. So I started from the very obvious thing. So I took the resting state data and trained the model from the very beginning. And I thought, how hard could it be? It's an advanced CNN. It should work. And as you can see, the data, the model predicted jumps that there weren't even there. And it was like using a sledgehammer to crack the nut. So I decided to focus on the loss function itself. The base model uses the Euclidean loss function, which heavily punishes the big errors and makes the model panics. So I created my own custom Huber loss function, which is great because it pays attention to small errors and does not freak out with the outliers. Thanks to the change of loss function, I managed to overfit my first model, and also that gave me a little bit overconfidence. So thanks to John's suggestion, I used the Ray library and decided to find the ultimate best hyperparameters to finally fine-tune this model. I thought that it would be it, but as you can see on the plots, as the trained data was amazing, the Pearson correlation was almost 1. The test and validation showed the Pearson correlation 0, which meant that I, again, overfitted the model and it did not generalize. So, also to check if what I did had any sense and any meaning, I did the leave one out cross correlation. I did it with different parameters from both my custom loss function and with base Euclidean loss function. And as you can see, especially on the first plot, we could not get over the barrier of 0.15 mean of Pearson correlation, and the mean of the Pearson correlation was almost 0. So that gave us the idea that we can't just focus on the distance of our errors, but we also have to show how this data is going, the movements and the structure of this data. So I basically baked the Pearson correlation into the loss function, and I managed to overfit again my model. As you can see, it's a quite big overfit. And then the final breakthrough, I used transfer learning, and thanks to the overfitting I took the overfitted model, which I already knew how the eye looks, and then only trained it on the very last few layers. And it was amazing because I got, for the first time, the Pearson correlation in unseen subject above 0.5, which did not happen before. So it was a really big success. and it was in almost in half of the test subject. And then we wonder, is it better than the linear methods itself? So I used the script that I got from Jamal and compared my transfer learning method to linear method. And as you can see, in 7 and out of 12 coordinates, the model I trained was better, which was success. And what's next? I plan to further optimize the transfer learning pipeline and also try to train the model on separate eyes. So it is quite essential for it is quite essential for stroke data. And at the very end, a little bit about me, who I am. So I am already finishing my bachelor's degree and in bioinformatics, and I am also a president of student association of bioinformatics in Silico. And thanks to this association, I managed to take part in different projects. One of them was the Challenge Camda. It was about microbiome. And I am also taking part in the project about creating the pipeline to automate detecting the structure of stroke in mice from fMRI. So my interests kind of correlate with what I am doing right now. And the last things I managed to also take part in was summer school, neuroengineering summer school that was in Brno by CEST, where I, for example, managed to find out how I would look if I was bald, and I managed to pet the cockroach. And also I took part in Brain Hack when I met Jamal. And I also became back then a pigeon whisperer, as you can see on the picture, a few hours before the Brain Hack. And at the very end, I can say that I also like to travel and I love music. And that would be all. Thank you for your attention, and I hope you had fun during my presentation.
Well, many thanks.
Proficient use of meme.
Well, I think that it's important not only for this project, this type of analysis of data, yes?
Yes, if we can beat the linear models, we will use the outputs of this model as the base of eye movement predictions from the neuroimaging data. So this actually plays a critical role on how we proceed.
So I think that you are happy.
Sorry?
I hope that you are happy having such a...
Yes, she's quite committed. Yeah, I'm happy with the contributions and progress that she made.
Okay. So many thanks.
And why
so
they were eggs on the last slide? Like an egg package.
Eggs.
Yeah, eggs.
Thanks.
Like chicken eggs.
It was a cockroach.
Okay.
So now we have as well last speaker of this session is Anastasia Poltarak. Well, I don't know, probably Anastasia will join us, yes. Well, he's also from practically the same place as Zofia is, because she's also studying bioinformatics at Wageningen University. And as I understand, he is interested in virtual reality, computational modeling, and he, together with you, leads this association of bioinformatics students. Ah, it's well, okay. And here, Anastasia is working in our team, in team structural and functional genomics led by Tomek Kościółek. So, Anastasia, screen is yours.
Okay, thank you so much. Hello, everyone. I'm Anastasia, and I would like to talk with you today about a world of protein discovery and visualization. And I want to dive a bit deeper into protein discovery and visualization, from language models to 3D structures. This field blends AI with biology to design novel proteins and gain better insights into their behavior. And I want to talk a little bit about ESM models. Let's start with evolutionary scale modeling models, advanced protein language models trained on massive amino acid structure sequence and databases, numbering in the hundreds of millions. They process protein sequences as input and generate embeddings. It is numerical vectors, as you know, that encode evolutionary history, structural details, and functional properties for tasks like classification and prediction. And there are some different versions of ESM, like ESM second, ESM third, and ESM C, Cambrian. And we will start from ESM second. It's a family of transformer models, training through masked language modeling on over 250 million protein sequences. As input, it's a simple amino acid sequence. As output, it's per residue embeddings that capture co-evolutionary signals. They are really good for function prediction, not so good as AlphaFold, but really good, and secondary structure analysis as well. The second is some third. It's an evolutionary scales flagship generate models. It's a lot of parameters there, like 96 billion parameters, train on sequences, structures, and functions using tokenization and diffusion. As input, we have sequences, structures, or prompts, and output, we have novel protein complete with predicted 3D coordinates and functions, like gene ontology terms. And the last one, it's SMC, or Cambrian, as I said. It's a family of models with different parameters, like 300 million parameters, 600 million parameters, 6 billion parameters, trained on vast metagenomics data through two-stage scaling. As input, we have sequences. As output, per residue embeddings that match larger to SM second models, and a fraction of the cost. And last one, I want to say about ASM Fold. And ASM Fold, it's a structure prediction head to ASM second, generating 3D.
Probably, I have impression that maybe it's okay, but I have impression that you are looking at the same slide all the time, and you are talking about something what is not on this slide, but maybe
Yeah. I just want to add this information to this slide. Yeah, because I think it's really interesting, because SM fold is something like adding to the SM second, and it's, as I said, it's a structure prediction head to ASM second, generating 3D coordinate directly from the sequences. And it's 10 times faster than AlphaFold second. And we don't need multiple sequence alignments for this. And we, as output, we will have a ready-to-use PDB file with confidence score. And I personally, what I'm doing in Sano, I personally benchmarked in some case SMC, SM second, SM third. At Sano, I generate embeddings from CAF, dataset protein sequences, wrote validation scripts and build classifiers to predict CAF classes. CAF, it's something like protein topology folds like alpha, beta, and 4-level hierarchy, class, architecture, topology, and homology. The goal is to evaluate how well embeddings capture structural topology without full 3D prediction. The process of my work is like embedding extraction, pooling. I use min, max, and attention pooling, training simple classifiers, and accuracy evaluation. And as you can see, some certs mean pooling is delivered the best results, a straightforward test that underscores the power of richer training data. But I have one limitation because prediction of the CAF class is very simple problem, and I can say that it doesn't fully stress models on subtle differences, like, for example, functions. And it can be different for different tasks, like, as I said, for a function, I think it will be better to use attention pooling, for example. And what I can say, what I can do in the future is bring something interesting, like protein visualization. Visualization, it is my passion. I'm fascinated to compare, for example, ASM predictions with AlphaFold predictions to allow, for example, prediction from AlphaFold and from ASM, and use the VR sets for immersive viewing. This way, the topic of my previous seminar at Jagiellonian University, as you can see on the pictures. And I would like to run something similar at Sano to showcase deep VR visualization integration. And I want to talk a little bit more about my other interests, for example, and projects. I'm also a part of HG Space Systems, and we have our project. It names Gravity, and this project explores how bone cells respond to biomaterial scaffolds in microgravity to advance gravity-dependent tissue engineering, crucial for astronauts, health, and Earth-based regeneration. And I think it's really interesting because it won the little bit contest, and we now doing the tests with the cells in at Jagiellonian University, and we hope that the next year or in 2 years our cells will go to the space. And I'm also I'm a vice-president of the Student Association of Bioinformatics at Jagiellonian University, and I really like to organizing events for pupils, like Olympiads, camps, and conferences, as you can see there. And I really like to participate in different challenges, and that's all from me. Thank you so much.
Well, many thanks. I think that we have seen overview of 5 different research projects at Sano, and I hope that point of view, the results are interesting enough to think about somehow to present them to broader audience.
Yes, I have some comments, especially on the data science presentation over there. So you mentioned data science. So what was your input towards the project? Because I know that collecting wearable data is not easy, right?
Yes.
So how you deal with that? So did you approach? So what was your approach in terms of that?
So first of all, the data was in a form of the feed files. So we need to first have some kind of a tool to convert it to the CSV files to then understand the data better. So this was done using Java to make from feed the CSV files. And as it is for the data itself, there was a lot of data from this wearable Garmin's, but we decided to stick only, not for example, from the data from the trainings themselves, but more or from the wellness data. So we have, for now, we tried a CNN, but it didn't work pretty well. And now we are working with the residual neural network, because I thought that maybe the like adding the skip connections and information from the previous 3 days would be beneficial for us, and for now it is. So we are doing it in a form of a windowing problem. So we take 30 days from past, and then we take 3 days and
you move it.
Yeah.
Okay. So, but you use the Garmin API to extract the data. So how did you have access to the data on the model file?
So.
And also you only use four features, right? From all the data, because that
no,
was.
I have more features. Like I only taught like the main groups, so the sleep, the HR, but there is more into it. So we have, for example, I don't know, the time of the sleep or the phases of the sleep or so on. So in all of those groups, there is more features. And so the feed files we take from actually the Garmin Connect club. So everyone that has the Garmin should sign up there, then take the data. And then by putting it to the web app, it is done, the converting is done automatically. So, yeah.
So how this connects with, so because you mentioned data science and then you mentioned your work at the hospital.
Yeah.
So, and then you mentioned statistics over there. So then you mentioned robotics later on. So,
all different areas.
But are you willing to tackle any of the fundamentals data science problems, like data imputation, why you put that imputation in the models, how to overcome some of the limitations the machine has. So are you thinking about that? Because data science, from my perspective, is it's not about filtering the data and curating the data. For that, you can do other things. It's about, how you deal with the limitations.
So, for example, when we don't have enough data from a user, then we we are not using the deep learning methods themselves. We are using like manually written script, which is actually giving us the readiness score for these users, because there's a limitation when we don't have enough data. Then,
but are you thinking on dealing with that limitation, or you are just accepting, okay, this is a limitation I need to?
No, I did it. This is in the code, so it is working, and for now it is working okay. But as I said, like we need more users, so we need more data, and then I will maybe tune the model better for the residual neural network to have better predictions. Yeah.
So, but how do you connect that with your hospital work, with your future?
So this was it is a different area than the one that I'm doing. Like I'm more into the healthcare and maybe project connected to it, but like it surely like gave me a lot of information about, for example, web development and also dealing with the messy data, because it was very messy when after converting to the CSV files. and I need to perform also some data imputation, as you said, and yeah, it doesn't realign with my interests so well, but still it's nice to like get to know also
Yeah.
the other areas. Yeah.
I was just curious.
Yeah,
because
no,
you
of
mentioned
course.
data science, and data science is
Yeah.
so broad.
Yeah,
I was
yeah,
just curious of the sense of how it connects with all the other things that you like to do.
yeah. Thanks.
Thank you for your explanation.
Thank you. Any other questions, comments?
I have a question regarding this project with pilots. So, when it comes to the situations that you mentioned, like painting while being in the air, it is very often connected not with like general cardiovascular health in particular day, but for example with the ability of the body to adapt to, for example, accelerations, blood pooling to specific parts of the body. But as I understood, the data that you collect are mostly collected on Earth during rest and some kind of exercise. But how to take into account, for example, this changing gravity, which can be very influential when it comes to situations like painting and changing gravity.
So this we didn't did like yet. We are also planning on like understanding better the situation that you talked about. But for now, we just take the data and not from the training themselves, but like from the rest and so on, just like you said, from the sleep data. And we will for sure do it in the future, but we don't do it yet. Yeah.
So they have to take off their watches while they're at the plane?
No, they don't have to. But we didn't like, we don't take this data into consideration when,
Just
not yet.
it.
Yeah,
Okay.
not yet.
Okay. So, if there are no,
I think there is Olaf.
There's Olaf, yeah. Olaf.
Okay, so yeah, thank you for the interesting talks. Actually, they had a certain pattern to it, I think. All of these projects are young, many of these projects are data projects, and several of them experienced some of the, let's say, 101 errors that you have when you start a data project, that your data are messy, that you are prone to overtraining because stratification is one of the first things you have to do in order to do those things. And I was wondering whether there is any, let's say, exchange of these experiences within Sano, so that when onboarding a new data project, these kind of things are somehow propagated so that you know about things like confounding data, something about biasing by embedding, something about assumptions. So, for example, let's say in the case of protein-protein interaction, you are assuming that there is no post-translational modification going on in secreted proteins, which is a very bold thing to assume. And those things you have to take into account in some cases. And yeah, for the pilots, I don't know. There is a lot of talk about drugs and alcohol in pilots, so I don't know whether you have specific patterns that you can predict whether there is a certain kind of modus of the pilot and those things. I think there are a lot of common experiences here that would probably benefit from some kind of exchange platform for the data projects at Sano.
well, I think that I will try to tell something about the situation. So we, first of all, there was a kind of discussion at the moment when the project were formulated at Sano teams, and then I think that there is exchange of results inside Sano teams because if I'm not wrong, in for example in computational neuroscience, there are something like four this type of project are in Jan team. There are at least 2 projects in Tomasz Kościółek team. So it's exchange becoming is going on here on the 3rd floor of Sano from time to time. At least I can observe this, and I think that it is for the first time a public presentation of the results. So I think that after that there will be also some discussion between different teams, and the discussion which was here between Jose and Zofia, I think it was not just Zofia, yes?
Yes.
It was also example of, let's say, a kind of discussion related across, let's say, team boundaries. So this is what everything what I can, let's say, tell you. We have still 5 other projects of this type to be presented. I think that we will be able to do this in a month from now. So, you know, this presentation was a way to discuss this internal.
Okay, good luck.
Thank you. So if there are no more questions and comments, so first of all, many thanks to these 5 young ladies who were brave enough to present the results here. And I think that we need also to thank Gosia and who was taking care about organizing this, and Gosia and Magda who were pushing our ladies to be brave enough to present the results in the public way. So thank you very much. Well, and as I understand, it is recorded, so it will be available for your families. At least. I hope that you agree for publication with on Sano YouTube. So many thanks.
Thank you.